We'll see how this goes. Hello, people at home. Wish you were here. These people in here are all gonna only say smart things. Okay, so we're gonna do our cell respiration today. So yesterday we drew the little mitochondria and we labeled it, with, kind of its parts, but also with what step happens where. So now we're gonna kind of make those steps come alive. I'm sure that you're totally excited by this. All right, so I'm gonna draw my mitochondria. That was kind of a really loud marker noise. All right, so here's our mitochondria. Now, at some point, I'm gonna expand it, so just be aware if you're drawing with me. Or if you feel like your mitochondria from yesterday is large enough, that's fine. But we are going to be writing what happens here, what happens here, what happens here. It's going to take us, and this part we'll probably have to like expand because it's going to be too small. You'll be able to see it. So that's what we're going to be doing. So if you need to give yourself more space, do so. I don't tend to make a list with this one for some reason, but we can do that if we need to. I'm going to get black markers so that it's more visible in the video, and it'll be more visible to you too. Win-win. Okay, remember the formula for cellular respiration is exact opposite of photosynthesis. So you're going to require, what do we need to do this? What do our cells, now this is not plants anymore, only autotrophs, but it's us too. What do our cells need to do this thing? What do we have to have? So we need some oxygen. Got to breathe it in. We need to eat something. What is it? Glucose. So we need some oxygen. We need some glucose. And then what are we going to get rid of in the end?
Carbon dioxide.
We're going to breathe out our carbon dioxide, right? And we're also going to breathe out some water vapor. Yeah, water vapor. Ever notice that when you breathe on a mirror, it fogs it up, but when you use the hairdryer on the mirror, it don't fog it up? Like what's going on there? You're actually breathing out water vapor. A lot of that water vapor is water that's produced in cell respiration. However, you can also use that water in your cells, brand new spanking pristine water that's just been created. So kind of fun. So the process starts, sorry, I have to kind of make sure I'm not pulling off the board here. The process starts with glycolysis. So step one, and we wrote that yesterday. So there's step one, glycolysis. Okay, this is happening in the cytoplasm. And if you want to be technical, and of course I do, we actually call that the cytosol. The cytosol is the actual jelly. The cytoplasm is the jelly and everything that's in it, including organelles, which is kind of weird. You technically should have learned it as cytosol from middle school, but for some reason, all of our textbooks and everything else call it cytoplasm right up until you get to AP Bio, and they're like, ha, JK, that's actually called cytosol, and cytoplasm is that and everything in it. And it's like, well, why did you tell us cytoplasm from the beginning? What the heck? I think we could have learned cytosol just as easy. So glycolysis is happening in the cytosol or the cytoplasm. You can use them interchangeably in AP Bio. It's not going to be the end of the world. It starts with glucose, okay, and it has two phases. So I'm going to draw a little thing here. It's going to split up our glucose. So our two phases are the energy investment phase because it has to pay a little energy to play. So we have to put in 2 ATP. Use the energy that's connected in that ATP, and then we get 2 ADP plus our inorganic phosphate. Okay, so that's our energy investment. I'll write that over here. So basically, it's going to break the glucose apart, but it can't just break the glucose apart without having some energy to do so. Okay, so it has to have a little bit of something to get it going. Okay. And so it does that. And then it goes through a series of steps, and there's a couple of different steps there. If you look up glycolysis online, you look at an image of it, you're going to see like a couple of different names and things. You know, it's a process, but we don't have to know those. So I'm just going to go straight to what we do have to know. That way you don't have to learn extra stuff. And that is that when it splits it into 2 Now this is 6 carbon, right? C6H12, okay. So when it splits it into 2 3 carbon molecules, excuse me, it gets a little ATP from the, now that we've got it broken, remember it was a high energy molecule, so we kind of start getting some of that energy back, okay? So it goes, you get your ADP, so you're gonna phosphorylate an ADP, actually 2 of them, and you're actually gonna do 2 over here too. So what has happened, sorry, this is gonna be a 2 as well. So 2 ADP plus inorganic phosphate yields ATP, 2 ATP, and then same thing on this side. So what's happened is your glucose is broken into 2 3 carbon molecules, and so then those 2 3 carbon molecules are going through the same processes simultaneously. And so anything that happens on to this left one is gonna happen to this one on the right as well. And so that's why you're gonna see on these arrows that I drew here, the same thing happens on both of them, okay? Because the glucose was broken in half that we end up with 2 molecules doing the same. I'm going to try to see if this red marker will show up. We have here also an electron carrier. Now this is not, your electron is not excited by, I think you can see that. This electron is not excited by the sun, right? It's excited by the energy that was stored in that glucose. So that's how it's energized. All right. And so of course, if we had it happen over there, then it has to happen over here too. Notice it's very, very similar to the one in photosynthesis. Only difference is it doesn't have that P, which doesn't stand for photosynthesis, but it's kind of helpful that it's a P. Okay, when we finish up, we get 2 3 carbon molecules called pyruvate. And that's what we were looking for. Okay, so, all right, so glycolysis happens outside the mitochondria in the cytosol, or the cytoplasm, don't call it that. It has to invest a bit of ATP to break the glucose in half. After it does that, then it goes through its energy payoff phase, where it converts the molecule a few different times. But in that phase, it actually gets a total of 4 ATP, 2 from each side, and it also gets 2 NADH, electron carriers, okay? Winds up with a molecule called pyruvate. So now glucose has turned into pyruvate. Good? Pyruvates are a 3 carbon molecule where glucose was 6. All right. Our net energy from this, so our net product is 2 ATP because we had to use 2 to start it and we got 4, so, and 2 NADH. So that's what we get from glycolysis. And when I say net product, I don't count pyruvate because pyruvate is moving on and getting modified. But right now we have literally produced for the cell those 2 things. The ATP will be like carried off and used for whatever is needed in the cell. Like, you know, if you want to make a protein or you need to have a energy for one of your pumps in your membrane or whatever you need it for, your cell will be able to use that ATP. The NADH often is carried to finish this process, oxidative phosphorylation step at the end. But sometimes it doesn't really make it. We'll talk about that when we get to oxidative phosphorylation. Okay, now, any questions about glycolysis? All right, glycolysis is highly conserved, right? That means that glycolysis happens in almost all organisms, even our prokaryotes, even in organisms that live in environments that don't have oxygen, so they can't really do normal cell respiration. We still see glycolysis happening, so it's extremely highly conserved. Okay, so we got our pyruvates here. We got 2 of them. I'm just going to send 1 in right now. So here goes our pyruvate into the matrix. Remember yesterday I said, okay, this is step 1 and Krebs cycle is step 2, although I would argue it might be step 3. All right, this is why, because I'm about to tell you what I think is step 2. I don't know why we just kind of poo-poo on this step. It's kind of an important one. But Just keep in mind, I'm only sending one pyruvate through, and there are two. So everything that we do from here on is double, okay? And it does it simultaneously. Like both pyruvate would come at the same time, and all these things would happen at the same time. You would actually have glycolysis occurring all around the mitochondria, and a lot of pyruvate is coming in. All this is happening, at the same time, all the time. Kind of makes me tired to think about it. Like all my cells are doing this all the time. I'm surprised I even have any energy at all. Wait, it's creating energy. I don't know. Anyway, deep thoughts. Okay. Pyruvate is changed into something called acetyl-CoA. This is one of these terms, acetyl-CoA, that you will hear called so many different things, like acetyl-CoA or CoA, or all kinds of weird things. I call it acetyl-CoA. I don't really care what you call it, as long as you sort of know what it is and can sort of spell it. Okay, acetyl-CoA is what I call it. Acetyl-CoA is a 2-carbon molecule. How many was pyruvate? 3. So, bye. Right? Got rid of some CO2. Breathe that out. I just did it. All my cells just did that. I don't even know. That's so weird. Okay, so breathed out that CO2, or actually we didn't breathe it out yet, right? It has to get to the cytoplasm, and then it has to go to the membrane, and then the membrane, it'll diffuse across the membrane. CO2, it's small and nonpolar, it'll go right to the membrane, right? So it diffuses across, it gets picked up by a red blood cell. Red blood cell takes it to the lungs. The lungs offload it because of diffusion as well, and then you breathe it out, and that does make me tired, thinking about all that. That's a lot of work. Okay, you can't keep it though because it's toxic. Okay, so also from here, we get another electron carrier. Yay, but no ATP. I mean, I feel like that's a legit step. I don't know why they just ignore it. They call it a link reaction sometimes, or they'll just smash it into the Krebs cycle and call it part of the Krebs cycle or something, but it's kind of before the Krebs cycle. So anyway, here's the Krebs cycle. Krebs cycle is going to be right here. Acetyl-CoA is going to join in with another molecule. It's called oxaloaceate or something like that. You do not have to know that. Clearly, I don't even know it, so why would you have to know it? Okay, but acetyl-CoA joins in with that. As it goes around the Krebs cycle, there's the Krebs cycle right there. As it goes around the Krebs cycle, and remember, it doesn't move, it's just sitting there, it's just, changing and whatever, and then that that oxalate, whatever thingamajigger is being created, so that's why we call it regenerated, that's why we call it a cycle. So what it does is it breaks completely down. So it was 2-carbon, right? It breaks all the way down, and so we get 2 carbon dioxide out of this. So there's all the carbon dioxide. Hey, is that all the carbon dioxide? How many did we say was going to be produced? In the formula, how many? 6. And so I only see 3. Why do I only see 3? Because it's doubled. We got 2 pyruvates that do this. So in the end, it will be 6, right? Because both pyruvates will do that. But from 1 glucose, you will get 6 carbon dioxide. Does that make sense? Good? Okay, so meanwhile, I am not going to be the best biology teacher ever, and I'm not going to tell you which step each of these electron carriers, I'm just going to tell you how many are made total. Is that okay with you, or do you want each step? You don't have to know each step. You guys feel okay, all right. So we get 3 total NAD plus converted to 3 NADH. Okay, so there's 3 NADH. One of them is made here, one right here, and one on the regeneration. But I'm just going to stick them all right there because, shoot, I don't feel like writing them all over the cycle. Okay, we also get a new one we've never heard of before. This is fun. We get an FAD plus converted into, it gets 2 of them. FADH2, it's a double. It's like a double-decker bus carrying all those little electrons. Okay, and finally, we get an energy molecule. I'm going to put ADP. It can also be GDP. Okay. And that can just go, bye. ATP leaving. It's going to do some work. Go do work. Okay. So, so how many ATP then approximately would be created from one glucose in the Krebs cycle? Because I have one here, but how many? Two, right? So approximately. I want to stress that these are approximate numbers, right? It's not always going to be, 2 ATP. And actually, there's something about GTP is actually kind of a smaller energy requirement. So, you may make a GTP if you don't quite have enough of everything going good. Remember, this requires a lot of enzymes and inputs and stuff like that. And if it doesn't go just prime, then you might not get both of your ATP. It could happen. Okay, so whenever they just throw numbers at you and say it makes this many, this many, this many, those are estimates and averages. Okay, and also different organisms make different numbers. So kind of cool. Although typically you get some. So what you really need to know is it a small amount of ATP or a large amount? Small amount here, small amount here. We're about to get the large amount. Okay. All right, so that's Krebs cycle. So what do you get from the Krebs cycle? You get 3 NADH, you get 1 FADH2, and you get some energy, either in the form of ATP or GTP. Okay? Cool. Questions? This is quite simplified. So if you look up the diagrams, it's kind of fun to look them up, but if you look at the diagrams, you're like, what the heck? There's so many steps. There are so many steps, but it's okay. We don't need to know. All right, so we're going to finish it up with the third step. So I guess this is technically the second step. With the third step, which is going to occur right here. Now, I'm going to draw the little proteins right here, but then I'm going to blow them up over here, okay, so that we can look at them without them being all squished. All right, so basically it is an electron transport chain, exactly like the one in photosynthesis. Oh, let me say exactly. It's got 4 proteins instead of 3, but it's very, very, very similar. Same process, got a pump for your protons, and then so we've got an electron transport chain here, right there. And then we have some chemiosmosis going on too. So we have an ATP synthase right here, or wherever, but they're usually next to each other. Okay. And all together, this is called oxidative phosphorylation. Okay. All together, these ETC and this chemiosmosis or ATP synthase, it's just called chemiosmosis because the protons move through it. It's like chemistry moving through. So, okay. So I'm going to draw it bigger over here. So I'm going to blow this little area up. So let's see, I'm going to have a little membrane going on, my little inner membrane, okay? And I'm going to have my outer membrane. So this is my inner. You can probably figure that out. Okay, and this is the outer membrane. Gross, quit coughing. We talked so much yesterday, I was coughing all day. I'm going to move my little video so we can see this better. Oh, and by the way, this video will just go on Canvas. So if you're just want to binge watch cell respiration videos, put it up there for you. Yeah. Season 1 and 2, whatever. Okay. All right. So let's see. Let's put on our little ETCs. So here we go. We got some proteins here. By the way, whenever we did the blast lab, talked about this a little bit yesterday. Whenever we did the blast lab, I said you could do hemoglobin alpha, or if you can't find hemoglobin alpha, use cytochrome C, because almost all organisms have cytochrome C. Cytochrome C is one of these guys right here, a protein that's in the ETC, and almost all organisms have it. So here's our little adorable ATP synthase. So cute. Okay. So this is going to be oxidative phosphorylation. Oh my gosh, the word is so long. Could we have made it any longer? Probably. And now what happens is, let's see if I can get both of these in. There we go. Now what happens is these electron carriers, or coenzymes, as they're sometimes called, these electron carriers, coenzymes, all start converging at the ETC. So they're all transported to the ETC. Now this one over here in glycolysis sometimes has a little trouble getting there. Sometimes it moves in and does fine. Sometimes it actually gets picked up on its way by something else that can use that little energized electron. And so it never quite makes it in. But that's why, once again, the ATP formed isn't like an exact number. Like, sometimes there's a little fewer and sometimes there's more, and it depends on some things. Okay, so we used to say you get 2 from glycolysis and you get 2 from the Krebs cycle and you get 36 from the ETC. Really, you can get anywhere from 18 to like 45. So it's kind of a big range. So we kind of seeing textbooks now having kind of a range or just kind of saying approximately because it's kind of a misconception to know, say that's like think we know every time we have a glucose how many we're going to get. Okay, it depends on a lot of factors. So here they come. We've got FADH2, we've got NADH, we've got NADH, we've got NADH, got a lot of NADH. DH, okay, so these are enzymes. These are proteins that are able to split off the proton from the carrier. So we'll basically be converted like that. Your little proton will be kind of hanging out here. Your electron will be passed through the ETC. Okay, there it goes. And the proton will be pumped through. So super similar to the photosynthesis, right? Where you have the electron moving through the electron transport chain and the proton being pumped through. Right, so you'll start to build up a gradient here after you do this with all these guys. Okay, cool. And you also have some just kind of hanging out here waiting to be pumped through. That'll be important in a minute, but all right. So then we have, all these little electrons that are going through here. And there they all are. Okay, cool. So far, so good. What's going to happen next, do you think? According to what you know from photosynthesis when you have a proton gradient and ATP synthase. Yeah, you're going to go whoop. You're going to see chemiosmosis where the protons move through, and you're going to see a creation of, or what's it, phosphorylation of ATP, or phosphorylation of ADP to create ATP. Goodness. Okay, and it's going to be a lot, like a lot of ATP. Okay, so that's good, but you may see an issue. I see an issue, but then again, I already know it, so that's not fair. You may see an issue in the fact that we're going to have a traffic jam. You can only pass down enough electrons until you don't have any space for any more, right? We got electrons just sitting there. Like, what are they going to do? Just like leave? They can't just leave. You can't just have electrons running willy-nilly around the cell. That doesn't work. And you can't have any more passage of electrons down and pumping of protons if you don't get rid of these guys. What part of the formula have we not used yet? Huh?
Water.
We haven't seen water. We haven't seen oxygen. So we're about to bring in the oxygen and release the water. Okay? So oxygen actually is able to enter the mitochondria. And when it does that, it comes in here and it will pick up, it's O2 coming in, it will pick up the electrons and will bind to a couple of these free protons and it'll make some water. Okay, so this is the final electron acceptor. That is a big point for College Board. Okay, this blue part is what they talk about quite a bit, and it's because it's like super important. Like it is super important. So then what happens is when it picks up with a couple of these little guys here, a couple of these protons, these free electrons hanging out here, and I'm kind of like, where do I draw this? Because it's going to be a mess no matter where I do. Made some fresh water. Brand new water. Never been drank before. It's rare to get never been drank before water. It's kind of a gross thought. Okay. So I shouldn't say it's rare. I mean, there it is, but we don't get to drink that. Sad. Okay. So let's talk about this for a minute. Just get over it. So what happens if we don't have that final electronic filter? Sorry, just throwing things over here. Just ignore me. Well, you probably know. What happens if you don't have any oxygen? You die. How much time you got? Oh yeah, I think they say 6 minutes to brain death. You know, or just the beginning of brain death. You can stave it off. I've heard of some people not breathing for like 20 minutes, but I don't trust those people after they come back to life. I'm not sure exactly what's been happening there. Yes, this is why you need to breathe. This process right here. You don't need to breathe because your lungs are like wanting some air. You don't need to breathe because your bloodstream needs some oxygen. You need to breathe because your mitochondria have electrons building up, and you got to get rid of them, or else your ATP process shuts down. And if your ATP building process shuts down, you die rather quickly. Okay? So, I'm sorry. Yes, I felt the same way. I started throwing things too. It was a little, sorry, that was a little harsh. So basically, I actually kind of enjoy thinking about this, though it kind of scares me a little bit. I'm not going to lie. That I kind of like knowing like what's actually going on. Like, so when you're working out, right? Or running from a bear, which is the only time I ever run, then I start to breathe more, right? But not because my lungs are like breathing more. Like, why am I breathing more? Like, what the heck? Because it's not because you're running. Like, that's not physically making you breathe more. Something in your cells is making you breathe more. Okay, and it's basically the fact that you're needing to do this process faster, right? You need more ATP. Your cells, your muscle cells are depleting the ATP. You don't have enough. Your brain uses so much ATP. Like, you know, all those pumps and things that are in your neurons, they got to run those all the time. And remember, it's like 700 miles an hour. And as soon as it goes, it has to go again. And so your brain is just like eating up the ATP. And then, of course, your muscles, anytime you contract, or I guess not relax, relaxing doesn't require it, but anytime you contract your muscles, that's a lot of ATP. So here you are running, and you're thinking about the bear, and you're kind of freaking out. All this is like ATP, I need it. And so what do you have to do? Get more oxygen, okay? Your heart's got to beat faster. Why? It's got to get the oxygen to where it's going, right? That's its whole purpose, is to like, let's move this blood through here to get that oxygen to where it's going to go. And that's why you breathe more when you exercise. It's not because of some exercise like magic that makes you breathe more. It's because you don't have enough ATP to continue doing what you're doing unless you breathe faster. And if you don't breathe faster, then you run out, and then you just got to like lay down on the ground and pant for a while. I mean, I kind of feel like that when I walk up those stairs. I don't know about y'all. I'm like, well, that was enough exercise for one day. About ran out of ATP in my brain. So that's kind of, I mean, that's the whole idea, right? I mean, that's it. That's why you're breathing more. That's why your heart's pumping more, because you got to get ATP and you got to get oxygen in there to pick up the electrons, or else you die, and that's tragic. Okay, so the other thing that goes with this is maybe you've heard of something called carbon monoxide. So while CO2 can be toxic at high levels, I don't know if you've noticed this, but if you're in a smaller classroom, ours is pretty good because it's so big. And plus, you know, see those little hearts hanging from the ceiling? Those are all fresh air vents. And I have like 6 of them, 1 over each table. And so I like to see if they're like have any air coming in. That's why I have hearts hanging there. So our room gets fresh air. Okay, it's rare for these classrooms to get fresh air, right? Unless you have a window or a vent like that. But if you have a small classroom for like 6th hour and you're in there, you might notice you get a little sleepy in 6th hour. And that's because you have a lot of carbon dioxide buildup in those classrooms, because hour after hour, students have been breathing out carbon dioxide until the levels start to go up. When they hit about 1,500 parts per million carbon dioxide, people start like nodding off because carbon dioxide can be toxic, right, to your cells. But Carbon monoxide is toxic really fast. Okay, so hang on a second, Shannon. I'll get right to you. So carbon monoxide is toxic to your cells really fast. So what it does, here's what carbon monoxide does. Carbon monoxide is just CO instead of CO2. It seems like such a small difference. Like, what the heck? But what it does is it goes into this protein here that has the electrons, and it binds to it. And now oxygen can't pick them off. And so now you're getting the same effects as if you just stopped breathing. Like, if you just stop breathing, your cell is going to have that same effect, because that's going to shut this down, which is going to shut down the Krebs cycle, which is going to shut down the pyruvate, and the glycolysis will go for a while. Like, it's like, I got this, guys. Like, I'm going to make some ATP for you. I know that we're about to die, but I'm playing my violin as the ship goes down. But then it can't. It can't do any more. It does as much as it can. But the whole reason is because carbon monoxide, there's a binding site on this that it binds with, and it shuts down that process. So then you have a matter of minutes before all of your cells are starting to have this problem. So here's my PSA for you, public service announcement. If you have any gas in your home, if it's attached to gas at all, natural gas or propane, either way, get a carbon monoxide detector and make sure it's like floor level. Okay, carbon monoxide is heavy, it sinks, and it doesn't smell. So it's kind of one of those sketch things where it's like, ooh, we all feeling really sleepy. Let's pass out on the floor. Bad idea. That's where the carbon monoxide is. So if you're gonna pass out, pass outside. Okay, so but that's why, because it's going to shut down your process, and you're going to make ATP. And if you don't have ATP, your cells can't do anything. And so then they just die. Shannon, do you have a question?
Is that why, like, you can't leave a car running with the garage closed?
Yes, because it is gas. So carbon monoxide is an off product of gas, basically burning gas, or in the case of natural gas, like stoves, like your stovetop, that's like natural gas, if it's not burning right, or if it's left on, not burning, so it'll burn kind of funky, and it'll have this off. It should burn it completely. Like, it shouldn't have any carbon monoxide release from any gas source in your home. But if something's wonky about it, it can. Or if it's releasing extra gas, not enough. Gas fireplaces, that have a gas, can also do the same thing. But all you need is a carbon monoxide detector, and they're very efficient. And you just put them in the same room with whatever gas source you have, and also gas heaters, same idea. But you put it down on the floor, which always freaks me out whenever I go to Lowe's and they're like, here's a smoke detector, aka carbon monoxide, 2-in-1, save money. And I'm like, well, do I put it on the ceiling or the floor? Like if I put it on the ceiling, it'll get the smoke, but carbon monoxide down there. Oh, so I would buy separate ones and put the carbon monoxide one down on the floor. So anyway, that's my PSA. Yeah.
What about lactic acid?
So actually, all of your dreams are going to be fulfilled when you watch the Ed Puzzle tomorrow, because it's all about fermentation, lactic acid buildup, alcohol fermentation. Well, it's basically glycolysis, like pushing through. But then also what about organisms that don't live in an oxygenated environment, right? Like, what about them? They have to do something. So it's got that on there as well. And I'll take you through the process of why it can't continue forever, and all that stuff. So yes, that is episode 2, season 4. I don't know what season 1 now. Okay, let's see. Anything else? Okay, so now what we're gonna do in our last 15 minutes is we're gonna go through it again. You know how we do. So you're gonna draw it. We're gonna talk through it. Make sure you've seen this process from glycolysis to oxidative phosphorylation at least twice before you leave my classroom. Okay, so get out whatever you do. Like, I don't know if you do. I mean, I don't want to just go through it again by myself. I don't need to do it. I've done it like 12, 15 million times. But if you want to draw it out on a whiteboard, or if you want to just kind of whatever, but you are going to go through it again. And then we'll have a quiz over this on Friday. We're going to go over it Thursday, but Thursday also we're setting up our Pansy lab, so we won't have the whole entire hour. So make sure you're reviewing it at home and studying it at home too. Okay? Bye, people at home.